Welcome students. I'm Dr. Kim Rodriguez, Assistant Professor in Botany at Kamal College of Arts, Science and Commerce for Women, Nuvengoa. Today I will be teaching Somaclonal variation for third year BSc Botany Semester 5 students. Paper code BOD 101, paper title Plant Tissue Culture. Title of the unit Unit 3 Cellular Totipotency and Differentiation. Module name Somaclonal Variation. Module number 18. The module outline is Somaclonal variation, types, causes, isolation, induction, methods of assessment, applications, and limitations. The learning outcomes of the module are defines the term somaclonal variation, explains the types, causes, isolation, induction, methods of assessment, applications, and limitations of somaclonal variation. Larkin and Scowcroft proposed the term somaclonal variation. Somaclonal variation are unexpectedly rich and novel changes among cells of callus cultures of somatic explants, which result in genetic variability in regenerated plants. Somaclonal variants or somaclones are plants exhibiting somaclonal variations. Example, aneuploids, sterile plants, and morphological variants, sometimes involving traits of economic importance in case of crop plants. Terms such as protoclonal and gametoclonal are used to describe variations from protoplast and anther or pollen culture, respectively. Types of somaclonal variation. There are two types of somaclonal variation: genetic variation and epigenetic variation. Genetic variation is a pre-existing variation in somatic cells of explant, whereas epigenetic variation is the variation which is generated during tissue culture. Genetic variation is heritable, whereas epigenetic variation is non-heritable. Genetic variation is stable and is transmitted to the next generation, thus important for crop improvement, whereas epigenetic variation is temporary but persists throughout the life of the regenerated plant only. The causes of somaclonal variation are changes of mother plant origin, explant-derived variation, genetic changes arising in culture, growth regulators, composition of culture medium, culture conditions, and effect of the genotype. Changes of mother plant origin. The variable chromosome number in different cells of a particular tissue in a mother cell may cause somaclonal variation. Explant-derived variation. Tissue systems differ in composition and physiological functions. As plant cells mature, their ploidy levels tend to change, thereby resulting in variation. Genetic changes arising in culture can cause somaclonal variation. Growth regulators such as auxins and cytokinins bring about karyotypic alteration. They trigger high rate of cell division and induce mutation. Composition of culture medium. Microsalts, chelating agents, metals, undefined substances present in the culture medium can destabilize the normal genetic makeup, thereby causing somaclonal variation. Culture conditions such as duration of culture, frequency of subculturing, temperature, and light affect the cell cycle, hence causing somaclonal variation. Effect of the genotype. Physical and chemical environment of the cultured cells cause stress on the cell, which is expressed by changes in cell population. Isolation of somaclonal variants. Somaclonal variations occur spontaneously in low frequencies. They can be screened and beneficial ones isolated. Isolation of somaclonal variants can be done by screening and cell selection. For screening, the mutants can be easily isolated from cell cultures. For cell selection, mutants can be effectively selected for disease resistance, improvement of nutritional quality, adaptation of plants to stress conditions such as saline soils, low temperature, resistance to toxic metals such as aluminium, resistance to herbicides, increased biosynthesis of important plant products, which can be used for medicinal and industrial purpose. Induction of somaclonal variation. Somaclonal variation can be induced by application of pathotoxins, herbicides, salts, metabolic inhibitions, or temperature shocks. The rate of variation expected in tissue culture is approximately 10 to 15%, which is very high in comparison to in vivo systems. Induction of somaclonal variation is a cheap and quick method of inducing variations. The regenerated plants can be tested for heritability, expressivity, and stability, and subjected to field trials. Methods of assessment. The cultures can be tested for genetic stability or variation. The parameters include phenotypic parameters, physiological parameters, and genetic parameters. Phenotypic parameters comprise of quantitative and qualitative parameters. Quantitative parameters such as leaf size, plant height. Qualitative parameters such as branching pattern, flower color, which can be analyzed. Physiological parameters. In physiological parameters, the protein patterns can be analyzed by electrophoresis. Formation of secondary products such as steroids or alkaloids can also be analyzed. Genetic parameters. The chromosome number and structure, banding pattern of chromosomes, alterations in DNA segments can be also analyzed. The applications of somaclonal variation are: new alleles or mutations may be isolated, which were not available in the germplasm or through mutagenesis. Single gene mutation in the genome may give the best available variety in vitro, having a specific improved character. Various cell lines selected in vitro are potentially applicable to agriculture and industry. The limitations of somaclonal variation are: somaclonal variation are applicable only to those species which regenerate complete plants. Selected cell lines often show reduced or no regeneration potential. Many selected clones show undesirable features such as reduced fertility, reduced growth, and even reduced overall performance. So to summarize, plants regenerated from tissue culture display somaclonal variations, which may be either genetic or epigenetic variations. Isolation and selection of somaclonal variants can be done by analysis of phenotypic characters, cytological studies, gel electrophoresis, and selection for resistance to diseases, herbicides, and environmental stress. The major benefit of somaclonal variation is to create variation in adapted genotype without the treatment of mutagenic substances. These are some of the references. Thank you.